Hello students, in this recording, we will see homologous recombination protein machines. That is all the proteins involved in the homologous recombination in E. coli. So all the proteins involved in the homologous recombination in E. coli, and in E. coli, there is a major double strand break repair pathway, which is called as REC-BCD pathway. So we have seen in the previous recording the double strand break repair model for homologous recombination and the steps involved in the same. Now we will see the proteins involved in each of the steps in E. coli. So if we see the first step of a double strand break repair model, there is introduction of double stranded breaks. Now there is no specific protein that has been found that carries out this task in E. coli. Rather, breaks generated as a result of DNA damage or failure of a replication fork are the major source of events in chromosomal DNA in E. coli. The first enzyme system that we are going to see in E. coli in this pathway is REC-BCD. DNA molecules with single-stranded DNA extensions or tails are the preferred substrate for initiating strand exchange between regions of homologous sequence. So if I say that in the double-strand break repair pathway, we have seen in the previous recording as well, that the first step is the introduction of double-stranded break, for which there is no specific protein involved in E. coli. After that, we have said that an enzyme comes, sits at these broken ends, and processes these broken ends to generate single-stranded DNA tails ending with 3'. We have seen in the previous recording. This processing at the broken ends to generate single-stranded DNA tails with 3' ends is done by REC-BCD. Because as we have said that DNA molecules with single-stranded DNA tails are the preferred substrates for initiating strand exchange between regions of homologous sequence. The REC-BCD enzyme processes broken DNA molecules to generate these regions of single-stranded DNA. So this step, what you can see in the double-stranded break repair pathway, after the introduction of the double-stranded breaks in one of the DNA molecules, this processing, what you can see to generate this single-stranded tails with 3' ends is done by REC-BCD. Let's see how REC-BCD does this. REC-BCD also helps load the REC-A strand exchange protein onto the single-stranded DNA ends. REC-BCD has many functions. So one of the functions is to process these broken ends after the double-stranded break to generate DNA with single-stranded 3' tails. But the other function is also to load another protein called as REC-A, which will be involved in the strand exchange, which we'll see later, onto the single-stranded DNA ends. In addition, the multiple enzymatic activities of REC-BCD provide a means for cells to choose whether to recombine with or destroy DNA molecules that enter a cell. So REC-BCD can decide whether to recombine with the DNA to undergo DSB repair pathway or the REC-BCD pathway, or to destroy the DNA molecules and does not recombine with them that enters a cell. We'll see this also. REC-BCD is basically composed of 3 subunits: REC-B, REC-C, and REC-D. These are the products of the REC-B, REC-C, and REC-D genes. And this REC-BCD has both DNA helicase and nuclease activities. DNA helicase, as we have seen, and as you know, you have done DNA replication, a helicase enzyme unwinds or unzips the DNA. So this is for separation of the strands, and this activity is for degradation of the DNA. So it binds to DNA molecules at the site of the double-stranded break, and it tracks along the DNA using the energy of ATP hydrolysis. It uses the energy of ATP hydrolysis because it's a helicase, and as a result of this action, the DNA is unwound. with or without the accompanying nucleolytic destruction of one or both of the DNA strands. The activities of REC-BCD are controlled by specific DNA sequence elements known as chi sites. In the E. coli DNA, there are DNA sequence elements known as chi sites. This is after the Greek letter. These sites control the activity of REC-BCD. How? We will see that. And this chi sites for crossover hotspot instigator. REC-BCD enters the DNA at the site of the double-stranded break, as we have seen, and it moves along the DNA, unwinding the strands. The REC-B and REC-D subunits, out of BCD, the B and the D subunits are both helicases. That is, they use ATP hydrolysis to melt DNA base pairs or unwind or unzip the strands. The nuclease activities of REC-BCD cleave each strand during unwinding and thereby destroy the DNA. So REC-BCD has both helicase and nuclease. With helicase, it unwinds the DNA, and with nuclease, it cleaves the DNA strands and destroy the DNA. So you can see here, there's a diagram, and this is one of the DNA molecules in which the double-stranded break would take place. After the double-stranded break takes place, like I have shown you in this step, if I go back to one of the previous diagrams, that the first step is the double-stranded break. Sorry. And after this double-stranded break, the REC-BCD will come here and use its helicase activities to unwind it, and also uses its nuclease to generate this single-stranded tails. So here also in this diagram, you can see that after the double-stranded break has been generated, you can see this REC-BCD coming and unwinding the DNA using the energy from ATP hydrolysis. And you can see these pieces, not only it's unwinding, it's also degrading the strands. But you can also see there is a green color part in this DNA sequence. And this green color part, what you can see is the chi site. You can see this green color sequence they have shown in this picture is the chi site, and REC-BCD will enter the site where the double-stranded break has been generated. It will unwind the DNA using the energy from the ATP hydrolysis because it's an helicase. It will start degrading the DNA strands. But when it encounters this chi sequence, its activity is changed. You can see in this step, the REC-BCD has encountered the chi sequence. Upon encountering the chi sequence, the nuclease activity of the REC-BCD enzyme is altered. And now, as REC-BCD moves into the sequence distal to the chi site, with respect to the broken DNA site at which the enzyme entered, it no longer cleaves the DNA strand with the 3' to 5' polarity. You can see in this step as well that it stops cleaving one of the strands. So it no longer cleaves the DNA strand with the 3' to 5' polarity. So one of the strands is not cleaved. Furthermore, after the encounter with the chi site, the other DNA strand, which is one with the 5' to 3' polarity, is cleaved even more frequently than it was prior to the chi site. The summary is that after the double-stranded break repairs are generated, REC-BCD will enter the site. It's a helicase and a nuclease. It will unwind the DNA using energy from the ATP hydrolysis. It will start cleaving both the strands. But once it encounters the chi site, it no longer cleaves one of the DNA strands, but it cleaves more frequently the other DNA strand, and this results in the generation of these single-stranded DNA tails, what you can see in this step, these tails. As a result of this change in activity, a duplex DNA molecule is converted into one with a 3' single-stranded extension, terminating with the chi sequence at the 3' end. This structure is ideal. The single-stranded DNA tail structure is ideal for the assembly of the next protein in picture, which is Rec-A, and initiation of the strand exchange. The molecular basis of the change in Rec-BCD's enzyme activity after the encounter with the chi site is unclear, but appears to be associated with either inactivation or loss of the Rec-D subunit. ssDNA tail generated by Rec-BCD must be coated by the Rec-A protein for recombination to occur. So now the single-stranded DNA tails have been generated. The next step will be strand invasion. The protein which is involved in strand invasion is Rec-A. Now the Rec-BCD entered the broken double-stranded break repair site and has processed the ends to generate single-stranded DNA with 3' ends or tails. Now the next step is strand invasion, which is done by Rec-A. So the single-stranded DNA tail generated by the Rec-BCD must be coated by the Rec-A protein for recombination to occur. But you know your basics that cells have SSBs as well, which are single-stranded DNA binding proteins which exclusively bind to single-stranded DNA and involved in replication and many other events. But here, SSBs should not bind. Rec-A only should bind for recombination to occur, and to ensure that Rec-A binds rather than SSB, these ssDNA tails, Rec-BCD interacts directly with Rec-A and promotes its assembly. So to ensure that SSB doesn't bind and Rec-A only binds to these tails, Rec-BCD directly interact with Rec-A and loads it onto the DNA and promotes its assembly. Chi sites increase the frequency of recombination about 10-fold. The ability of chi sites to control the nuclease activity or Rec-BCD also helps bacterial cells protect themselves from foreign DNA that may enter via bacteriophage infection or conjugation. The chi site, if you ask me the length of this sequence, it's 8 nucleotide sequence. The 8 nucleotide chi site or sequence is highly overrepresented in the E. coli genome. It's there in E. coli genome at many places. Whereas it's predicted to occur only once every 65 kb or 65,000 base pairs, or about 80 times, the chromosomal sequence of E. coli reveals the presence of 1009 chi sites. And because of this overrepresentation, E. coli DNA that enters an E. coli cell via conjugation or any other genetic transfer, that is likely to be processed by Rec-BCD in a manner that generates 3' single-stranded DNA tails and thus activated for recombination. So recombination will occur or promoted if E. coli DNA enters an E. coli cell because the chi sites are there. In contrast, if DNA from another species, let's say bacteriophage or any other organism in which E. coli chi sites are not overrepresented and will lack frequent chi sites, Rec-BCD on this DNA will lead to its extensive degradation and it will be destroyed rather than activation, and no recombination will take place. So this was the first protein in this Rec-BCD or DSB repair pathway of E. coli, which is Rec-BCD. Let's study the second protein in this major DSB repair pathway or Rec-BCD pathway of E. coli, which is Rec-A. You must have heard of this Rec-A protein. Rec-A is the central protein in homologous recombination. It is the founding member of a family of enzymes called strand exchange proteins, and it's one of the major proteins of this pathway. These proteins catalyze the pairing of homologous DNA molecules. And when I say pairing of homologous DNA molecules, it involves both the search for sequence matches between the two molecules and the generation of regions of base pairing between these molecules. So this is one of the steps of the DSB repair pathway. So if I say the first step of a DSB repair pathway is introduction of double-stranded breaks, which for there no specific protein has been found in E. coli. Then processing of those ends to generate these 3' single-stranded tails. This is done by Rec BCD. Then Rec BCD loads Rec A onto these tails. Then what you can see in this step, what we have seen in the previous recording as well when we did DSB repair pathway, that this single-stranded DNA tail pairs with the complementary strand in the other duplex molecule, and this strand which is peeled away or separated from this unbroken DNA molecule, this is gonna pair with the other strand or other tail. And this strand invasion, what you can see on this slide, this is done by Rec A. Active form of Rec A is actually a protein DNA filament. Unlike most proteins involved in molecular biology and genetics that function in smaller discrete protein units, such as monomers, dimers, or hexamers, Rec A filament is huge and variable in size. Filaments that contain approximately 100 subunits of Rec A and 300 nucleotides of DNA are common. So, after the Rec BCD generates these 3' single-stranded DNA tails, and then Rec BCD loads Rec A onto these tails, there are around 100 subunits of Rec A on these tails, and 300 nucleotides of DNA are involved, and these are called as active protein DNA filament, which are gonna do this strand exchange. The filament can accommodate 1, 2, 3, or even 4 strands of DNA, and you can see in this step how there are 3 strands involved here. Filaments with either 1 or 3 bound strands are most common in recombination intermediates. The structure of DNA within the filament is highly extended compared to either uncoated ssDNA or a standard B-form helix. On average, the distance between adjacent bases is 5 angstrom rather than the 3.4 angstrom degree spacing normally observed. Thus, upon Rec A binding, the length of a DNA molecule is extended approximately 1.5 fold. It is within this Rec A filament that the search for homologous DNA sequences is conducted and the exchange of DNA strands executed. So, when the Rec BCD loads the Rec A onto these tails, and these tails base pair with the complementary strand in the other duplex molecule, they will search for homologous sequence and then do this strand invasion. To form a filament, subunits of Rec A bind cooperatively to DNA. Rec A binding and assembly are much more rapid on single-stranded than on double-stranded DNA, thus explaining the need for regions of single-stranded DNA in strand exchange substrates. The filament grows by the addition of Rec A subunits in the 5' to 3' direction, such that a DNA strand that terminates in 3' ends is most likely to be coated by Rec A. Note that in DSB repair model for recombination, it's DNA molecules with just this structure that participate in strand invasion. So if I take you back to this diagram, the Rec A will bind onto these tails, and these tails will do strand invasion, because once the Rec BCD loads Rec A onto these tails, there are some 100 units of Rec A and some 300 nucleotides of DNA involved called as protein DNA active filament. And when this protein DNA active filament pairs with this complementary strand and do the strand invasion, they search for homologous sequences. And what happens is that These are the single-stranded DNA tails that are involved in or participate in strand invasion. Rec-A catalyzed strand exchange can be divided into distinct reaction stages. First, the Rec-A filament must assemble into one of the participating DNA molecules, which we have seen. Assembly occurs on a molecule containing a region of single-stranded DNA, such as ssDNA tail terminating with 3' end, I've shown just now. This Rec-A ssDNA complex is the active form that participates in the search for a homology. During this search, Rec-A must look for base pair complementarity between the DNA within the filament and the complementary strand of the new DNA molecule. Once a region of base pair complementarity is located, Rec-A promotes the formation of a stable complex between these two DNA molecules. And this Rec-A bound 3-stranded structure is called a joint molecule and usually contains several hundred base pairs of hybrid DNA. It is within this joint molecule that the actual exchange of DNA strands occurs. And the DNA strand in the primarily binding site becomes base paired with its complement in the DNA duplex bound in the secondary site. Strand exchange requires breaking of one set of base pairs and the formation of new set of identical base pairs. So if I take you back to again the same diagram, here you can see that the blue strand is being peeled away or separated from its complementary blue strand, and the gray strand, which is coated with Rec-A, is searching for a homology and pairing with this blue strand. So one strand is separated, melting of base pairs, and the other strand is pairing with the complementary strand in the unbroken DNA molecule. Breaking of one set of base pairs and formation of new set of identical base pairs. Completion of strand exchange also requires that the two newly paired strands be intertwined to form a proper double helix. Rec-A binds preferentially to the DNA products after strand exchange has occurred, and the binding energy that actually drives the exchange reaction towards the new DNA configuration. So we have completed the role of Rec-A as well. Now, after Rec-BCD and Rec-A, we move to the next proteins in this pathway, which is RUVAB. After the strand invasion step of recombination is complete, the two recombining DNA molecules are connected by a DNA branch, the X-shaped structure known as a holiday junction. These holiday junctions should migrate. Movement of the site of this branch or migration of these holiday junctions requires exchange of DNA base pairs between the two homologous DNA duplexes. Cells encode proteins that greatly stimulate the rate of branch migration. So, now we have come to this step. So how the two DNA molecules align in the DSB repair pathway, that is the first step. Then the double-stranded break takes place in one of the DNA molecules with the other remaining intact. There's no protein found in E. coli for that. Then the processing to generate 3' single-stranded DNA tails, that is done by Rec-BCD. Then Rec-A is loaded, that is also done by Rec-BCD. Then the next is strand invasion and searching for homology, that is done by Rec-A. And after the repair and joining, you have this structure with two holiday junctions. Now, these holiday junctions should migrate, which is the branch migration. And this moving of holiday junctions or branch migration is done by the RUVAB proteins. So RUVA is a holiday junction specific DNA binding protein. It's a specific DNA binding protein, but it's a holiday junction specific DNA binding protein. So RUVA will only bind where there is holiday junction. So if you can see in the previous slide, there are two holiday junctions. RUVA is going to bind to both of them. RUVA is a holiday junction specific DNA binding protein that recognizes these holiday junctions or the structure of the holiday junction regardless of its DNA sequence. RUVA protein is not bothered by the DNA sequence. Regardless of the DNA sequence, it can recognize a holiday junction structure and can bind to it. RUVA recognizes and binds to the holiday junction. What next? Not only it recognizes and binds to holiday junction, it's going to recruit another protein, which is RUB, to this site. So RUVA recognizes the holiday junctions, then it recruits RUB protein, and RUB is a hexameric ATPase. That means it hydrolyzes ATP. It's similar to the hexameric helicases involved in the DNA replication, and the RUVB ATPase provides the energy to drive the exchange of base pairs that move this DNA branch. So the RUVB basically is going to migrate these holiday junctions, continuous melting of base pairs and joining of base pairs. The last protein is RUVC in this pathway. Completion of recombination requires that the holiday junction between the two recombining DNA molecules be resolved or resolution. In bacteria, the major holiday junction resolving protein is RUVC, which is an endonuclease. You know the difference between an exonuclease and an endonuclease. Okay. RUVC is an endonuclease. That means it can make cut anywhere in the middle of the DNA. RUVC was discovered and purified based on its ability to cut DNA junctions, which are made by REC-A, that is these holiday junctions, in vitro. So it was discovered, it was purified based on its ability to cut these holiday junctions, which are made by REC-A in vitro in the lab. Genetic evidence indicates that it functions in concert with RUVA and RUVB. Now we come to this step. You can see that how these holiday junctions migrate with the help of RUVB. This cleavage of these junctions and the resolution into the two DNA molecules, this is done by RUVC. Resolution by RUVC occurs when RUVC recognizes the holiday junction, of course, likely in a complex with RUVA and RUVB, and it knits two of the homologous DNA strands that have the same polarity. This cleavage results in DNA ends that terminate with 5' phosphates and 3' OH groups that can be directly joined by DNA ligase. So ligase is also involved. We have seen in the previous recording, depending on which pair of strands is cleaved by RUVC, the resulting ligated recombination products will be of either the splice crossover or the patch non-crossover type. Despite recognizing a structure rather than a specific sequence, RUVC cleaves DNA with modest sequence specificity. Yes, RUVC recognizes a structure rather than a specific sequence. But despite recognizing a structure rather than a sequence, RUVC still cleaves DNA with modest sequence specificity. Cleavage takes place, what we have seen in the previous recording, DSB repair pathway, whether it's at site 1 at both junctions or site 2 at both junctions, or 1 at X at site 1, Y at site 2, or vice versa. This cleavage takes place only at sites conforming to the consensus sequence, which is first nucleotide is either A or T, second nucleotide is thymine, third is also thymine, and the fourth one is either G or C. Cleavage occurs after the second T in this sequence, and sequences with this consensus are found frequently in DNA, averaging once every 64 nucleotides. So this consensus sequence is found in DNA once every 64 nucleotides, and this modest sequence selectivity ensures that at least some branch migration occurs before resolution, because without this sequence selectivity, RUVC will come at the holiday junctions and will simply cleave the holiday junctions, both at site 1, both at site 2, or 1 at site 1, site 2, as soon as they are formed, thereby restricting the region of DNA that participates in the strand exchange, because the cell doesn't want RUVC to cut the holiday junctions as soon as they are formed. That's why a slight delay should happen, so that the more region can participate in strand exchange. RUVC cleaves with sequence selectivity after the second thymine. So we have seen in this recording the detailed role of the proteins involved in the DSB double-strand break repair homologous recombination pathway in E. coli, also called as REC-BCD pathway. We have seen REC-BCD, we have seen RUVA, RUVB, RUVC, and of course, before that, REC-A. Now, to conclude this recording, to summarize it, we are going to see the DSB repair pathway diagram again, and we are just see that which protein is involved at which step. So the first step in a DSB repair pathway where the two chromosomes align, and then a DSB break takes place in one of the chromosomes with the other remaining intact. No protein has been found in E. coli for this step. Next is processing to generate these gapped DNA with 3' single-stranded tails. These tails. This step, processing to generate single-stranded tails with 3' ends, is done by an enzyme which has both helicase and nuclease activities, that is REC-BCD. And REC-BCD also loads REC-A, the next protein in picture, onto these strands before SSB can bind onto this. Then this strand invasion, what you can see, this REC-A, which is coated on these tails, and the REC-A protein filament has formed. It invades the strand in search for homology, and then this strand invasion on this slide, what you can see, is done by REC-A, and the active is protein DNA filament. Next is the RUVA protein, which recognizes and binds to these holiday junctions. After that, RUVA recruits another protein, RUVB, which migrates these holiday junctions or branch migration. And the last step, this cleavage, is done by endonuclease RUVC, which will cut both at site 1 or both the junctions at site 2, or one junction at site 1, one at site 2, and depending on that non-crossover, crossover products will be formed with sequence selectivity. This is RUVC. In this recording, we have seen all the proteins involved in double-strand break repair pathway in E. coli, also called as REC-BCD pathway. REC-BCD, REC-A, RUVA, RUVB, and RUVC.